Okay, so let us start with this organelles, Golgi apparatus. Okay, so we look at Golgi apparatus. What we need to know in terms of the structure, again, they are single membrane bound organelle. Right, single membrane bound organelles consist of flattened sac again. Right, consists of flattened sac. Okay, it's a membrane single membranous sacs or termed as cisternate. So what's the difference between Golgi apparatus and the Aria? You look at Aria, they are interconnected. Okay, Aria, they are interconnected. So the cisternate is an interconnected cisternate. But for Golgi apparatus, not interconnected. Okay, most of the time they are not interconnected. And then you look at this Golgi apparatus, they are formed by a stack. Okay, a stack separate internal space from the cytosol. So what's the difference between cytoplasm and cytosol? When you say cytoplasm, basically organelles together with the solutions of the cells. Are you clear? So when you use the word cytosol. So cytosol together with the organelle form cytoplasm. Okay, cytosol together. So sol here means solution. So it's the solutions of the cytoplasm together with the organelles form cytoplasm. Okay, so it means that this cisterna, okay, form a stack that separate the internal space from the cytosol. So as I said before, it formed this what we call the compartmentalization. And we give them the name. So when you see this symbol indicate the extra information for you guys. Okay, so you look at the stack, then we can identify one is a cis and now another one called transverse. It's just a name given to them, right, to show that's a slight difference in terms of their structure. Generally, where is a cis phase? Cis phase going to face or near to the RER. Okay, so therefore the transport vesicle move the material, for example, modified protein, or we also term it as the RER product. Right, received from who? From the Aria to the Golgi apparatus. So it means that the vesicle that bud off from the Aria now can add the membrane to the contents of the lumens at the cis phase by fusing with the Golgi membrane. Okay. Then the Golgi apparatus dispatch its products by budding the vesicle from the trans phase. So it means that they're going to form this what we call the Golgi vesicle or secretory vesicle at the trans phase. And then the transverse face what? Transverse generally faces the cell membrane. So that the vesicle now will move toward the plasma membrane, fills with the membrane, and then the molecule are released out of the cells by the process called exocytosis. So exo means out, send out. Cytosis, send out of the cells is known as exocytosis. Take it in from the cells. Okay, take it into the cells is termed as endocytosis. The opposite one is called endocytosis. Send it out by exocytosis. Okay, so before we continue with the cis and trans phase, now you can actually look at this diagram. You realize that this is the cis phase. So cis phase is the one that receives the transport vesicle. Okay, then you can see the trans phase. Trans phase is the one that can you see that the bud off and then form the secretory or Golgi vesicle. Okay, so I give 5 minutes, label the diagram, and then I'll disclose answer. Okay, so I assume that you guys have done the labeling here. So what's the first structure here? So this is a transport vesicle, okay, from Golgi, from the Aria. And then the membranes of the transport vesicle fuse with the membranes of the Golgi vesicle at the cis phase. Okay, so transport vesicle, let's say for example from rough ER, okay, or you label as a transport vesicle enough. Okay, so you can see that this is a cisternate, okay, cisternate of the Golgi apparatus. They are not interconnected, but the diagram show here interconnected. I will explain it later why this is interconnected. Okay, so the second part here is the cisternate, or you say flattened sac, both accepted. Okay, and then we can see that the vesicle, okay, start to bud off, correct or not? Okay, so new forming, right? A new vesicle forming or the new forming vesicle. Okay. So this vesicle now bud off, okay, from the transverse. Okay, secretory or Golgi vesicle from transverse bud off, and the last one, okay, secretory or Golgi vesicle. Okay, so means if you put it here as the bud off and this one is the Golgi vesicle, both accepted. Okay, so how we know it's cis phase or transverse? Basically, cis phase is the part that receive the vesicle. Okay, they receive the vesicle, so bud off from the transverse. Okay, so I'm going to show you the drawing, how we draw the Golgi apparatus relative to the rough ER and also the plasma membrane. Okay, so we do have this rough ER. They're interconnected, rough ER. How we know rough ER? So I put some ribosome here, okay? Some ribosome. Okay, then how I will draw the Golgi apparatus? So when draw the Golgi apparatus is something like an arch. Okay, you draw it something like an arch. Okay, arch this way. Okay, so basically the cis face here facing the cis face faces the rough ER. So this is the rough ER, and the transverse facing what? Facing the south membrane. Okay, this is a cell membrane or plasma membrane. So what will happen here is you can see that after the modification inside the rough ER, so rough ER going to pack this product in a transport vesicle. So they pack it, and then this vesicle bud off, continue, move, and then it fuse, and then they release the content into the cis phase of the Golgi apparatus. Then inside here, we have some processes take place. And then sometimes you may see that, how come they connect? Sometimes. It's not all the time they connect. Okay, so why they connect? They connect so that this item or chemical or molecule can move from one compartment to another, from one cisterna to another cisterna, and then they call it back. So they are not interconnected permanently, unlike the Aria interconnected permanently. So this vesicle at the ends, okay, coming here already. So packed as the Golgi vesicle. So this Golgi vesicle will move towards, can you see that? They're going to move towards the cell membrane and then attach to the cell membrane. Can I see the fuse to the cell membrane? So when it fuse the cell membrane already, then what you can see here is, now look at this. When it fuse, so it forms something like this. And then it can release the protein, okay, glycoprotein out of the cell. So by the process called exocytosis. Okay. Okay, you want to take a photo? You can. This is how the process, why the Aria are in between the nucleus and also the Golgi apparatus. And then why Golgi apparatus, the cis phase face the Aria? Because the transport vesicle from Aria will fuse with the cis phase of the Golgi apparatus. And then through the some activities, after that we pack them in the Golgi vesicle, and then we carry out exocytosis to send out the, okay, this for example glycoprotein. Okay, okay. So now look at this. A quick revisions, as I say that in the Aria, what we do in the Aria, we have the folded, folding of the protein, okay, polypeptide into the precise three-dimensional shape. And second, inside Aria, we also do the glycosylations. And third, we actually join the protein or assembly, okay, of the polypeptides to form the complete protein. Okay, so once we have done already, we pack them, right? We pack them into the transport vesicle. So what is the content of this transport vesicle? The content inside this transport vesicle is termed as Aria product. A product of the rough endoplasmic reticulum. So what are the possible Aria products? It can be a complete protein. It can be a complete protein. It can be a glycoprotein. Okay, it can be a glycoprotein. So this Aria product will be received. Okay, received by who? Received by the Golgi apparatus. Okay, so what Golgi apparatus is going to do? Golgi apparatus have two rows. Okay, so Golgi apparatus, the first row, further modified. Okay, further modifications of Aria product. Now, what means further modification of the Aria products? So involve what? The first process here involve the further glycosylation. What is further glycosylation? Yesterday we learned about this, right? So this is the protein, for example, and then we already add, okay, short fragments of the sugar. Okay, let's say we add sugar. Okay, can I see that? Okay, we add. So this, the whole thing is known as the glycoprotein. Okay, it's termed a glycoprotein. So first process, further modification, means that what we're going to do here, okay, in the Golgi apparatus, okay, I think that we need to add more. Okay, we add more sugar here. We can add, complete the add. Okay, and we can remove, oh, this part we have to remove. So enzyme are going to help us to remove and then maybe transfer to here. So it's going to make our protein more variety now. Okay, make it more variety of the glycoprotein. Can I see that? From initial, then we have the further modifications or further glycosylation, make them into different form of the glycoprotein. Okay, and this is particularly crucial, okay, for the cell recognition. Okay? Glycoprotein involved in the cell recognition. So how the cells actually know? Like for example, our immune cells, for example, lymphocyte, how the lymphocyte actually know, oh, this is our own cell, this is the bacterial cell, this is the fungus cell, this is not our own cells. So how they can recognize? They recognize the glycoproteins, okay, as the surface antigens. Okay, this is the first. So once they finish the process, everything already, so Golgi apparatus are going to do packing. Okay, so pack what? Pack this Golgi apparatus product or called Golgi product into Golgi vesicle, so that this Golgi vesicle later can carry out exocytosis. Okay, so two major functions of the Golgi apparatus: further modifications of the Aria products. We continue to make more variety. Okay, and then we also pack them. Okay, so it's something like sorting center, sort them together, because they come in, you may have enzyme 1, enzyme 2, enzyme 3. So we pack them nicely together, and then we can carry out the exocytosis. How we pack them? We pack them in the Golgi vesicle. Okay. So let us look at this, the functions of the Golgi apparatus. So you can see that this is the rough ER, right? Okay, rough ER. How we differentiate this rough ER? Because of the ribosome, the dots, and also interconnected sac. Can I say that? Interconnected sac or cisternate. So this one is smooth ER. Tubular structure. Okay, so you can see that the vesicle bud off from the Aria. So this vesicle contains the Aria product. So Aria products going to be accepted by the Golgi apparatus at the cis phase. This is a trans phase because it's nearer to the plasma membrane. Can you see that? Nearer to the plasma membrane. So after the further modifications, can you see that? So when finish a modification, we're going to pack them into the Golgi vesicle. So inside this, we can send them out. Okay, so what are the products? Okay, again, what are the products inside this Golgi vesicle? It can be proteins or glycoproteins. Are you clear? It can be proteins or glycoproteins. Okay. So describe two functions. So we look at the first functions. So polypeptide modifications. Okay, polypeptide modification. So basically, Golgi apparatus removes some sugar monomer and substitute other to the modified polypeptide, which is the Aria product, producing a large variety of carbohydrates on this glycoproteins. Okay. And second row involved in what? Involved in the sorting, packaging, and send out the Golgi products. So at the transphase of the Golgi apparatus, the Golgi product are sorted, packaged into several different transport vesicles. For example, it can be Golgi vesicle, it can be lysosomal vesicle. So these vesicles are sent to their respective area of the cells, or actually can send it out by exocytosis. Okay, so these are the role of the Golgi apparatus. So if you look at the diagrams here, in diagram 1.16, so how you differentiate Golgi apparatus, can you see that? They form an arch like this, the arch. So where is the cis phase? So cis phase. This part transphase, can you see that with the Golgi vesicle, Golgi vesicle. Okay, can you see that here, cis phase? This one is the transphase. Okay, so Golgi vesicle are formed. Okay, so with this, we have done for the Golgi apparatus.